So I chose Escherichia coli, which is better known as E. coli, for my term project. It is a bacterium that typically lives in the intestines of healthy people and animals. Majority of the strains of E. coli are naturally harmless or may cause brief diarrhea and vomiting, such as E. coli HS, UTI 89, and CFT 073. However, there are a few strains such as E. coli O104H4, which is also called enterohemorrhagic E. coli, that caused an outbreak in Europe in 2011. Now, according to the CDC, the latest outbreak back in 2022, it was due to E. coli O157H7, a strain that has the symptoms of severe stomach cramps, bloody diarrhea, and vomiting, has been the cause of at least one foodborne outbreak in the U.S. each year since 2006. Now, the anatomy of E. coli, this is my hand-drawn picture. You have the outer membrane, the cell wall, the periplasmic state, the plasma membrane, the pili, which surround the bacteria, the cytosol, the DNA, the ribosomes, and the flagella. Some characteristics of E. coli are that it is gram-negative, it is rod-shaped, non-spore-forming, motile with paratrichous flagella or non-motile, grow on MacConkey agar, and can grow under aerobic and anaerobic conditions, and E. coli does not produce enterotoxins. Now, there is a such thing as positive E. coli that I had mentioned earlier. Now, E. coli naturally lives in our intestines as well as the intestines of most warm-blooded mammals, as it helps maintain the balance of normal intestinal flora against harmful bacteria and synthesizes some vitamins. The vitamins primarily synthesized by positive E. coli in our intestines are vitamin K, which supports blood clotting, bone metabolism, and regulates blood calcium levels, and vitamin B12, which supports blood cell formation, cell metabolism, and DNA production. The letters and numbers that name a strain serve as a code for which sugars are present, while the sugars the bacteria display aren't what make you sick. They're quickly and easily detectable and help scientists and doctors differentiate whether a present strain will generate toxins that can make you ill. Now, some of the primary causes for the infections, as 70,000 cases are reported to the CDC each year. Some of the causes for those are undercooked hamburger, swallowing a mouthful of contaminated pool water, person-to-person contact, unpasteurized milk that can carry E. coli due to a cow's udders and contaminated equipment, runoff from cattle farms contaminating fields where fresh produce is planted. Spinach and lettuce are especially vulnerable. And cattle being slaughtered and E. coli in their intestines can get on the meat and can cross-contaminate with other meats. There was an outbreak in 2022 that took place between July 26th and August 17th of 2022. This primarily affected romaine lettuce. 109 people were infected with the strain E. coli O157H7 across 6 states. Of the 97 people with information publicly available, 52 were hospitalized and 13 developed hemolytic uremic syndrome, which can cause kidney failure, but no deaths were reported. 82 people detailed their food history, with 68 saying they ate at a Wendy's a week prior to symptoms. Of the 68 people, 46 reported eating romaine lettuce. Now, as a result of this, Wendy's removed romaine lettuce from their sandwiches in the states affected, which were Indiana, Kentucky, Michigan, New Jersey, Ohio, and Pennsylvania. And this was thought to have been caused by polluted ground and surface water where the romaine lettuce was being grown. Those affected and the leading causes are young people and other adults. Those who with weakened immune systems, such as the people who have AIDS, those who are taking drugs for cancer treatments, or prevent the rejection of organ transplants. Time of year, especially between June and September, see higher infections, eating certain foods, and unwashed hands. And prevention. Always cook hamburger to an internal temperature of 160 degrees Fahrenheit. Avoiding unpasteurized juice and dairy products, preventing cross-contamination, washing raw produce thoroughly, proper hand washing, and if you have diarrhea, you should not prepare food, bathe with others, or swim in public pools or lakes. E. coli may have a bad reputation, however, majority of the time it is healthy bacterium. Whether it is helping our body produce vitamin K or vitamin B, E. coli has a purpose. Fortunately, we can prevent further infections and further outbreaks from occurring. Proper food preparation and hand washing are two of the primary ways to help with these. Ensuring we take the proper steps to contain the contamination of food and the spread of bacteria due to us never truly knowing if we are infected is vital to prevent the unnecessary spread of bacterium. And here's my bibliography.
This is my term project. It is on Bifidobacterium, and I'm just going to be using pictures from my graphic novel I made to explain what Bifidobacterium is. So in this drawing here, I'm using Bifidobacterium to represent basically a group of warriors, a part of a kingdom that fight, protect, and gain resources for the kingdom, which would be the human body, the kingdom. And Bifidobacterium makes up 90% of gut microbiota in infants in the early stages of life, and slowly dwindles down as you grow older, of course. And the most prevalent bifidobacteria in infants were B. longum and B. breve, and they are, as I said, the most abundant in the whole gut microbiota. So the role of Bifidobacterium is to ferment lactose and oligosaccharides, which are complex carbohydrates. They basically metabolize HMOs, which are non-digestible in the human system, but because Bifidobacterium exists, it metabolizes those, and it will help boost immune system. It will help digestion and prevent pathogens from attacking. So let's go to the next picture. Bifidobacterium, let's talk about the shape and anatomy of it. It's a gram-positive bacteria. It is basically a rod-shaped, branched looking bacteria. It's an obligate anaerobe, a lactic acid producer. It's also uses non-digestible plant polymers for energy, and it has irregular borders and has a white convex. It does not produce any spores, has no capsules, and is non-motile. It also comes from the Actinobacteria phylum, which I mentioned in the third picture there, or the first picture on top. And it is very good at growing in environments from 36 degrees Celsius to 38 Celsius, which is perfect in your stomach. Along with that comes lactobacillus and bacteroids, which are prevalent in the early stages of life as well. They also help ferment lactose. As you can see, they're buddies next to each other. And essentially, what Bifidobacterium does in the environment, like I said earlier, it surrounds the intestinal tract with itself and prevents pathogens from attaching to the intestinal tract. It also not only produces lactic acid, but produces acetic acid as well, which reduces things like yeast and molds in the gut. The bacteria itself is very not non-pathogenic. There's maybe one strain out there that can possibly be pathogenic, but it's very, very minimal. So as we grow older, though, bifidobacterium naturally decreases because just as we get older, it just does. And so, here I have a picture of the kingdom itself rapidly declining, representing the human body, decaying over time. That's just how life works. And right here, although they dwindle in population, they still do their job or try to do their job, but because as we grow older over time, enterobacteria and other pathogenic bacteria will enter the body, onto the gut microbiota, and have more opportunity to attack, basically. And in the end, although they're, low in population, they still try to do their job and do their best at what they do, which is protecting the body and trying to ferment lactose.
Hello class, this is a term project on cyanobacteria, also known as blue-green algae. So cyanobacteria have inhabited the earth for an incredibly long period of time. Early cyanobacteria are credited with playing a role in the Great Oxidation Event, where oxygen became a major component in Earth's atmosphere some 3 million years ago. Different species of cyanobacteria can take on different physical characteristics, as you can see in the photo on this slide. For example, some cyanobacteria can be unicellular, filamentous, or colonial form. However, all cyanobacteria use sunlight as an energy source, and they do this via photosynthesis. Photosynthetic pigments inside the cell absorb light as a source of metabolism, and oxygen is the major byproduct. Cyanobacteria can essentially be categorized as aerobic photosynthetic prokaryotes. But since this genetic range of cyanobacteria is so big, I chose specifically for this project to hone in on one specific species of cyanobacteria. That specific species is Prochlorococcus. Prochlorococcus kind of gained my interest because it was, and I quote, "It is possibly the most plentiful genus on Earth. A single milliliter of surface seawater may contain 100,000 cells or more. Prochlorococcus is ubiquitous in our oceans that lie between 40 degrees north and 40 degrees south latitude, and plays a pivotal role in marine ecosystems, food, and biochemical cycles. Prochlorococcus has a coccus shape and is very small, 0.5 to 0.7 micrometers in diameter, making it the smallest photosynthesizing organism in the entire world. It is single-celled and is a prokaryote that possesses a mixture of chlorophyll A and chlorophyll B. Cyanobacteria in general have cell walls composed of peptidoglycan. However, Prochlorococcus have cell membranes composed of sulfolipids instead of phospholipids, enabling it to survive in nutrient-poor water. The thylakoid membranes that you see in the pictures here are the site of light capture, and it's where photosynthesis actually happens. The pigment lies within the thylakoid membranes, and it's in these which photosynthesis occurs, releasing oxygen as the byproduct. Let's look at Prochlorococcus's habitat a little closer. Here in this beautiful picture, we have a chunk of ocean and the sun in the upper right corner. Remember, we said our friend Prochlorococcus likes to hang out between 40 degrees south and 40 degrees north in our oceans. It mainly likes this zone because of its preferred water temperature. This specific zone is called the photic zone, where the sunlight and UV radiation can still penetrate through the water and photosynthesis can still occur. This is how they provide the Earth with nearly 2/3 of our usable oxygen. So for context, our forests contribute about 1/3 of our usable oxygen. So it creates almost 70 to 60% of our oxygen that we have here on Earth, which all of the organisms can appreciate. So this photic zone extends approximately to 650 feet deep, which means all of our Proclochus are hanging out in these zones. And one interesting thing that I read about is that once the Pleurococcus finds the preferred habitat, cells will multiply and cultivate within a certain depth below the surface. And these different populations that will hang out at different depths have a name because they will multiply and cultivate within their own different populations. And these different populations are called ecotypes. And these different ecotypes will have certain populations of Proclochus that only want to hang out within these certain depths in the ocean. And their form and function will be dedicated to carrying out their photosynthetic duties at these specific depths.
Caulobacter crescentis by Ezra King. What is C. crescentis? C. crescentis is a bacteria known to be one of the most sticky natural substances and can be utilized for medical purposes. To put this into perspective, C. crescentis can withstand about 5 cars worth of weight due to its sticky substance. Caulobacter crescentis can be utilized as a biosensor for uranium, a bioremediation tool for heavy metals, an anti-tumor immunization technique, and an antiviral microbicide in humans. C. crescentis is non-pathogenic, so it is safe for humans, but further research is being conducted to ensure that this is true. Characteristics of C. crescentis. C. crescentis is a gram-negative bacteria. It has a fusiform bacilli shape. This is different from a regular bacilli in the fact that its cell body is more elongated and thin, but it still does maintain an oval shape. Depending on the stage, the bacteria has either a stalk and a holdfast, flagella and pili, or stalk and pili. Stalk and holdfast acts as a fastener to hold onto biotic or abiotic surfaces. Length of the stalk depends on amount of phosphate present in the environment. If there is not enough phosphate present, or a lack of phosphate is present, then the stalk will lengthen to increase phosphate taken. When there is a lack of phosphate, the stalk can appear to be as long as 30 plus nanometers in length. When there is sufficient phosphate in the environment, the stalk grows to be only about 1 to 2 nanometers. Where does Caulobacter crescentis reside? It is an oligotrophic bacteria, which means it resides in fresh water, lakes, streams, or rivers. Generally, oligotrophic areas are very nutrient-poor environments. Because C. crescentis resides in such environments, slow growth, lower rates of metabolism, and low population density is expected. Life cycle of Caulobacter crescentis. C. crescentis begins as a motile swarmer cell, and it is important to note that it is unable to undergo DNA replication during this phase. It is easy to identify whether or not the cell is in its motile swarmer cell phase because there is a clear flagellum attached. The purpose of this flagellum is to add motility and allow the cell to swim to areas that are more rich in nutrients. After it finds a nutrient-rich environment or it's satisfied, which would be an environmental stimulus, or when it's ready to develop, which would be a developmental stimulus, it will eject the flagellum and retract the pili. The stalk and holdfast then replace the bacteria. The holdfast is at the tip of the stalk and is what has chemicals that create this sticky substance. Once it has become a stalk cell, it is ready to replicate. During the pre-divisional phase, the daughter cell will produce a flagellum. And once the daughter cell detaches, the cycle repeats. It is important to note that a protein known as the CTRA develops a flagellum once phosphorylated. On the other hand, DNAA allows for DNA replication by unbinding the double helix. DNAA is unable to cause replication when CTRA is active because CTRA does the direct opposite and inhibits DNA replication. That means that naturally, there will be more CTRA in swarmer cells and DNAA in stalk cells. All in all, C. crescentus is an amazing bacteria because of its potential for medical uses. Because of its unique structure and ability to replicate its unique anatomy, C. crescentus can help scientists discover more about the bacterial world and its applications to the biomechanical field. Thank you.
This is the term project on Enterobacter. Enterobacter is a bacteria. Some familiar pathogens are Salmonella and E. coli. Enterobacter infections are serious infections with a high mortality rate, even with appropriate treatment. It can be found on human skin, plants, soil, water, sewage, intestinal tracts of animals and humans, and dairy products, and clinical specimens such as feces, urine, blood, and wound openings. Enterobacter species are responsible for causing many infections and less commonly community-acquired infections, which are urinary tract infections and many others. Enterobacter is a genus, and they are motile, non-spore-forming, rod-shaped cells, some which are encapsulated and has flagella. It also ferments glucose with acid production and reduced nitrates. Enterobacter thrive in both aerobic and anaerobic environments, which means that they are classified as facultative anaerobes. In the United States, from 1976 to 1989, it was found that Enterobacter was the third most common pathogen in the respiratory tract, the fourth most common pathogen in surgical wounds, and the fifth most common pathogen in the urinary tract and the bloodstream. Enterobacter can ferment both glucose and lactose as a carbon source, and gas is produced from the metabolic processes, but they do not produce hydrogen sulfide. Several Enterobacter species possess multiple plant growth promoting activities which exert their actions directly or indirectly. Enterobacter species are naturally susceptible to a range of antibiotics. They have natural antibiotic resistance. An increase in the use of antimicrobial agents within the hospital setting has led to the development of resistance to many beta-lactam antibiotics and beta-lactamase, with about 25% of Enterobacter generally growing well, aerobically and anaerobically, at temperatures ranging between 20 and 37 degrees Celsius. Antibiotic resistance is a growing problem with regard of treating the Enterobacter infections. Some of these possible treatments include beta-lactams, beta-lactamase inhibitors, and aminoglycosides. Enterobacter species are widely dispersed in nature and exist in a diverse range of environments, including soil, water, domestic, and food processing establishments, vegetation, vertebrate and invertebrate hosts, and in the feces of human and animals.
My name is Taylor. I'm going to tell you about Helicobacter pylori. H. pylori is a bacteria that's been infecting humans for 60,000 years, and it's estimated that half of all people actually have it, although most are asymptomatic. H. pylori is the bacteria that causes gastritis, peptic ulcers, and can even lead to gastric cancer. It has an interesting history of discovery. Dr. Barry Marshall and Robin Warren, they found that H. pylori was colonizing their patients' stomachs, and that when they treated them with antibiotics, the ulcers and the H. pylori disappeared. Dr. Marshall actually drank a culture of his H. pylori to test this theory and to show that H. pylori does lead to gastritis. And indeed, he did develop gastritis after an endoscopy and stomach biopsy. They found H. pylori in his tissues, and thus they confirmed their suspicions and eventually ended up going on to win the Nobel Prize for their discovery. H. pylori is a helical bacillus. It's gram-negative, has 4 to 6 unipolar sheathed flagella, microaerophilic, heterotrophic, relying on its human host for nutrients. And it is interestingly not an acidophile, even though it lives in the acidic human stomach, which we will explain momentarily. So how does it interact with its environment and other microbes? Well, H. pylori secretes urease, which is an enzyme that hydrolyzes urea into ammonia and carbon dioxide. This neutralizes the stomach acid so that it can survive. It has adhesins, which are outer membrane proteins, allowing it to attach to epithelial cells. It's got anti-phagocytic enzymes to stop it from being phagocytized, as well as a helical shape and flagella, which allow it to swim through mucus and burrow in there. This figure that I created of H. pylori depicts the outer membrane, peptidoglycan layer, cytoplasmic membrane, and flagella. You can also see the purple little dots representing adhesin proteins, little triangles representing the anti-phagocytic enzymes, and little squares representing the urease. So H. pylori also impacts the microbiota in the stomach by competing for nutrients, by changing pH, which can improve by raising the pH, it can create a better environment for microbial growth. It also influences gastrointestinal hormone secretion, for example, by stimulating the secretion of gastrin and ghrelin, both of which can influence microbial levels, and it also affects the immune response. So how does this impact humans on a larger level? Well, humans can develop gastritis, peptic ulcers, and also gastric cancer as a result of H. pylori. How it does this, at least with peptic ulcers, is by burrowing through the stomach mucus to the epithelial cells. It attaches to the membranes of the cells and multiplies there. It secretes exotoxins that trigger inflammation and thin down the layer of mucus. This exposes the epithelial cells underneath, allowing stomach acid to destroy them. Symptoms of peptic ulcers can be stomach pain, nausea, and heartburn. So to conclude, H. pylori is one of the most widespread but underappreciated infections in the world because it can alter the host physiology and even suppress the immune response for one's entire life. It is a group 1 carcinogen that has changed our understanding of disease causation because in the past, ulcers were attributed to stress, and now we know that it is actually caused by this bacteria, making H. pylori the first species of bacteria known to cause cancer. Thank you so much.
Hello, my name is Maham, and I chose to do my project on Rickettsia. So Rickettsia is from the family Rickettsiaceae. There are 4 genera. There's Rickettsia, Echerichia, Aurantia, and Cohexia. The genus I'll be focusing on is Rickettsia. The characteristics of Rickettsia are it is a small gram-negative bacteria. It is rod-shaped. It's very variable spherical bacteria. It is non-motile. It's an obligate intracellular parasite, and the outer membrane of the lipopolysaccharide is made of lipopolysaccharide with endotoxin activity. Its order is Rickettsialis, class of alpha proteobacteria, and phylum is from proteobacteria. So how does rickettsia actually get into the host? The bacteria infects an arthropod, which can be a tick, lice, flea, or mite that acts as a vector. The arthropod then infects a human and reaches the host cell. The bacteria can survive, grow, metabolize, and reproduce within the host cytoplasm. This is also where rickettsia can receive ATP for cell function. Once the bacteria has caused significant reproductive damage, the host develops an illness. Rickettsia reproduces through the means of binary fission. When rickettsia enters a host, they target smooth muscle cells and multiply by binary fission in the cytosol, which is the fluid portion of the cell cytoplasm, and causes heavy damage to the host cell. A reminder that binary fission is a form of bacterial asexual reproduction, where there is separation of a single microorganism body into two bodies, and therefore it divides genetic information into two parts. Metabolism of rickettsia is a little complicated. Due to its intracellular pathogenic nature, rickettsia can only survive in a host cell since it uses the host's machinery for metabolism and growth. Although rickettsia has limited function of its own metabolism, where it may produce ATP through oxidative phosphorylation. In the host, rickettsia uses the zipper and trigger mechanism to enter the host target cell. Rickettsia then uses the ATP-ADP symporter to exchange host ATP for bacterial ADP. Additionally, rickettsia membranes are permeable to NAD+, which is a host energy source. On its own, rickettsia uses blue oxidation to drive electron transport chain, which is coupled with oxidative phosphorylation in order to generate ATP. Since rickettsia cannot use glucose as a nutrient, they oxidize amino acids and citric acid cycle intermediates instead. Rickettsia's relationship with other organisms usually consists of invading the host and starts host colonization. After entering the host, rickettsia encounters host innate defense surveillance and may dodge many of them to colonize the host. The impacts of rickettsia are pretty severe. These bacteria cause rickettsiosis, which are disease caused by rickettsia, and most often cause acute febrile illness symptoms. The three most well-known illnesses from rickettsia are Rocky Mountain spotted fever, epidemic typhus, and marine typhus. Rocky Mountain spotted fever is caused by bacteria Rickettsia rickettsii and is transmitted from hard ticks among rodents and within rodents and humans. The patients present with fevers, headaches, and chills. Epidemic typhus is also called loose-borne typhus, which is mostly because the bacteria Rickettsia prowazekii is vectored by lice that infects humans. Patients are presented with high fever, depression, and rash. Finally, marine typhus from Rickettsia typhi uses rodents as their main reservoir. The rat flea transmits bacteria among animal hosts to humans. These symptoms include headaches, abrupt fever, and chills. So I decided to do my art integration project, which includes a drawing of how rickettsia infects and colonizes the host cell. In the first section, rickettsia is in a tick parent vector. Through the transovarial transmission, which is the process of a tick parent passes bacteria to its offspring. The offspring now is the vector of rickettsia. Rickettsia is replicated within the arthropod cell. In the third section, the arthropod has bit the human and has infected the human by breaking the skin barrier and is infecting the bacteria, and the bacteria infects the human cell. Rickettsia can now introduce itself into the host cytoplasm and cytosine, where it can reproduce cytoplasm and cytosol, where it can reproduce via binary fission. The cytosol of the cell is also the main area where rickettsia receive host ATP. Due to this metabolic process, rickettsia can survive, grow, and reproduce enough to cause disease to the human. Rickettsia is a very important bacteria for us to pay attention to since it causes significant human disease, and we should pay attention to them when we're traveling and when we are traveling in the United States in tick-infected areas so we can protect ourselves from acute febrile illness. Thank you for listening. Bye.
Hello, this is Zuan Wu. and I'm doing my presentation and project on Salmonella. So, what is Salmonella? So, Salmonella it's a common bacterial disease that affects mainly the intestinal tract. It mainly lives in animals and the human intestines, and usually shed it by, pooping. Most people get infected by either getting contact with water or food. Shapes and anatomies. Salmonella is genus of motile gram-negative and Patricus bacilli, I don't know how to say it, but it basically means it is rod-shaped. It is usually around 2 to 5 microns long and by 0.5, 1.5 microns wide. It's anaerobic and possesses three major antigens, that being H antigen, O antigen, and V antigen. The structures of Salmonella usually comprise of flagella, plasmid, DNA, capsule, cell wall, plasma membrane, cytoplasm, and pili. Spread and growth. So, Salmonella is spread via fecal or route and can be transferred by food, water, direct animal contact, and human contact. But that rarely happens, as most of the time, like nearly 94% of the time, is via food. So, how does it grow? Salmonella mainly grows via binary fusion, like E. coli. So, what they do is when you get affected, they go into your intestines and they attach themselves to the epithelial cell lining. When they're in, when they're inside, they begin to colonize it and reproduce. While it's doing that, they kill off the host cell in process, causing the many symptoms of salmonella, and then we'll move on to the bloodstream. Interactions with organisms and environment. So organs that are closely tied to salmonella are just mainly animals, such as cattle, chicken, rodents, reptiles, amphibians. These guys can carry the salmonella in their intestines and not even show one sign of the illness. So you have to be pretty careful around that. That's why sometimes your teachers may warn you about touching school pets, such as turtles, as you may get the salmonella. I remember this one time back all the way at middle school. Yeah, I had a teacher and she had a pet turtle. And she just had it in the classroom. And she put up a sign saying, "Do not touch, as salmonella is abound." So, environments. Salmonella are resistant enough to the cold temperature that allow them to not be destroyed when frozen, but things such as UV light and heat can kill them. No questions asked. So that's why they usually love wet environments that is shielded from the sun. So what are the symptoms of salmonella? Usually the incubation period of salmonella can be 6 hours or all the way up to 6 days. So, you never know. Possible signs, symptoms of salmonella or diarrhea, stomach cramps, fever, nausea, vomiting, chills, headache, and lastly, blood in the stool, which is not fun. I never had it, but it doesn't seem fun. I guess I can show you what I did for my project. So what did I do? Yes, one of these. Wait, no, that's the wrong one. God damn it, I messed up. But okay, what I did for the project was I made a comic. It's about a bunch of these. There's a bunch of people. It's basically like centered around a mafia. So big guy, big boss boy, and he messed with another moth boss. His name is Tony Lazzuto, okay? And you don't mess with Tony Lazzuto, yeah? So Tony Lazzuto sent best assassin, Sam Bellaella, short for, you guessed it, Salmonella. So he hires his two best people, Tony Two-Face and Ludovico Harchin, right? And their goal is to stop this Sam and make sure he doesn't kill the mafia boss. Alright, and I think that's it. Thank you for watching my thing. Whoever you are out there, good luck on whatever you're doing. Alright.
Clostridium difficile, aka C. diff, is the main culprit of a healthcare-associated infection that causes severe diarrhea. C. diff was discovered by Louis Pasteur in the 1860s and was first isolated from the intestinal flora of newborns in 1935. It was originally named Bacillus difficile because it was difficult to culture and study. C. diff is a gram-positive, obligate anaerobe, bacillus-shaped bacterium and has a vegetative form and spore form. Because it's an obligate anaerobe, it cannot survive in the environment for very long when it's in its vegetative state. When it's in its spore form, it is metabolically inactive and can withstand oxidative stress, extreme temperatures, desiccation, and acidic environments. The C. diff spores in the stool of those who are infected can live in the environment for extended periods of time, as they can survive harsh conditions, including the surfaces that are sanitized with alcohol. C. diff is part of the phylum Firmicutes and has many relatives, including Clostridium perfringens, which can cause food poisoning and diarrhea, and Clostridium tetani, which can be found in soil and cause spastic paralysis. When C. diff enters its host, it navigates through the acidic environment of the stomach and then travels to the small intestine where it germinates. For germination to occur, germinants and the proper nutrients must be present in the environment. Bile acids, along with glycine and other amino acids, trigger the germination of C. diff spores. Glycine and the other amino acids act as co-germinants. The spores then move into the anaerobic environment of the large intestine where they multiply and colonize. They then attach to the epithelium of the colon and secrete toxins that cause the infection. C. diff is found in soil, feces, sewage, and food and is transmitted through the fecal-oral route. CCPA is the main carbon catabolite regulator that also reduces the toxin gene expression. It uses the Stickland reaction, which oxidizes and reduces glycine and proline, which produce ATP and NAD+. When there's a limited supply of proline or proline reductase, alternative pathways are used to regenerate NAD+, which produces butyrate, which then stimulates the synthesis of the toxins of C. diff. Antibiotic use decreases the overall diversity of the intestinal microbiome. Specifically, broad-spectrum antibiotics decrease the amount of bacteroidetes, which allows germination and growth of pathogenic microorganisms such as C. diff. When the gut microbiota changes, the number of metabolites in the intestine are also affected, which alters bacterial fermentation. This decreases the amount of fatty acids while increasing carbohydrates and amino acids, which allows C. diff to use these metabolites for germination, colonization, and growth. Those who are at high risk for contracting C. diff infection include the elderly with multiple comorbidities who frequently undergo urgent surgery. Those who become infected with C. diff tend to be readmitted to the hospital, which provides the pathogen with more opportunities to spread to its next victims.
Azospirillum is a genus of gram-negative alpha proteobacteria. They are surface-dwelling bacteria living symbiotically with the host plant's root system. These bacteria convert atmospheric nitrogen gas into ammonium ions, creating a nutrient-dense soil and providing the plant with nutrients that are usually limiting factors for plant growth. 20 varieties of Azospirillum have been classified, but the most widely studied and used are Braziliense and Lipopharum. Braziliense is used in the production of grasses like sugarcane and cereals. Lipopharum is used in the mass production of corn, wheat, and rice. Azospirillum are rod-shaped bacteria with polar flagella made from glycoproteins and covered by a surface polysaccharide. Braziliense has hooked rods on the cell's polar ends of the bacterial cell wall. Individual cells have multiple membranous hooks around 75 nanometers long, anchoring itself to the roots of the host plant. Azospirillum stimulates production of phytohormones, polyamine, and trehalose. Phytohormones are organic molecules that affect seed germination, root growth, stem and leaf growth, flowering, and seed dormancy. Azospirillum synthesizes the phytohormones cytokinins, auxins, salicylic acid, abscisic acid, gibberellins, and ethylene. These bacteria are bioremediators, breaking down hydrocarbons and heavy metals. Azospirillum removes environmental iron using siderophores, synthesized molecules that combine extracellular iron with other elements, forming a complex that gets transported through the plasma membrane into the microbial cell. Siderophores lower levels of extracellular iron, stopping iron metabolism from unfavorable phytopathogens. The synthesis of metabolites promotes pathogenic resistance in the host plant, referred to as an induction of systemic resistance. Hydrocarbons are byproducts of industrial drilling and extraction released into the environment through spills, leaks, runoffs, and other industrial wastes. Heavy metals found in industrial fertilizer, mining, damming, and wastewater runoff seep into the soil and water. These xenobiotic contaminants cause huge shifts in the environmental microbiome and the microbial diversity present. The shift in soil chemical makeup allows microbes with adaptations to harsh climates to thrive, metabolizing contaminants as they flourish. This process is called microbial remediation. Azospirillum also induces antioxidant and enzyme synthesization in the host plant, promoting resilience to abiotic stressors and environmental increases in extracellular free radicals. Among abiotic stressors, salinity can cause drastic reduction in protein synthesis, photosynthesis, respiration, and nutrient absorption. Braziliense counteracts saline stress by limiting sodium ion absorption and promoting absorption of potassium and calcium by the host plant cells. In the induction of systemic resistance, Braziliense increases the plant's tolerance for salinity as saline inhibition travels from the part of the plant inoculated with Braziliense to the other regions of the host plant.
If the name itself didn't give it away, it is a coccus-shaped bacterial species arranged as a Staphylococcus. It is also gram-positive and halotolerant. It is a facultative anaerobe, so it primarily prefers an aerobic environment. However, in an anaerobic environment, it can ferment different sugars, primarily lactose when available. It is a low GNC content species, and it can produce catalase, which actually is unusual for a bacterial species that has a low GNC content. It is found in the environment and is a normal component of the human microbiome. Among other Staphylococcus species, one of note and one primarily found within the microbiome is going to be Staphylococcus epidermidis, where you will find Staph aureus within and on the human body would be in the nasal cavity and upper respiratory tract, moist skin folds, the gastrointestinal tract, and the urogenital tracts. Commensal bacterial species aid in the regulation of Staph aureus, Acinetobacter baumannii being one of note, along with species from Streptococcus and Corynebacteria, as well as other Staphylococcus species, which include Hominis epidermidis and Lugdunensis. A. baumannii is commonly found in wound cultures where Staph aureus is also present, and it actually exhibits strain-specific antagonistic characteristics towards Staph aureus, meaning that specific strains of A. baumannii will have very specific effects against specific strains of Staph aureus. It is pathogenic and can cause a number of serious and potentially fatal infections. Starting with skin infections, we have folliculitis, which is redness and swelling around one or a few different hair follicles, which can then spread into surrounding tissue or can spread deeper into different layers of the skin and into the muscle. Scalded skin syndrome, which is a product of exfoliative toxins produced by S. aureus that cause the skin to slough off, which then leaves someone in a very vulnerable state to secondary infections, which is where a lot of the problems and complications will arise. And then necrotizing fasciitis is a development of different skin infections from Staph aureus, specifically from methicillin-resistant strains. It is very rare and is known as flesh-eating bacteria. Bacteremia, which are bloodstream infections, are also very common with Staph aureus. Anytime you have a skin infection, you will take blood samples to see if it has infected the blood, and it can cause a number of serious syndromes, illnesses, diseases such as osteomyelitis, endocarditis, and toxic shock syndrome, which toxic shock syndrome can be fatal. Antibiotic resistance is very important when talking about Staphylococcus aureus. It is considered to be a superbug. Staph aureus developed resistance to penicillin, which was the first antibiotic discovered in the 1940s, very, very quickly, and roughly 80% of strains of Staph aureus today will be resistant to penicillin. In the late 1950s, around 1959-ish, methicillin was developed as a semi-synthetic form of penicillin, which could still work on Staph aureus. And by like 1960, 1961, resistance was detected in some strains of Staph aureus and is commonly known as MRSA or MRSA. And today, roughly 80 to 90% of strains will be methicillin resistant. And MRSA is not only resistant to methicillin, but also to macrolides, aminoglycosides, and cephalosporins, which are different types of antibiotics. Vancomycin very quickly became the antibiotic of choice around the 1980s, but very quickly in the 1990s, resistance was discovered in some strains of MRSA itself and is known as vancomycin-resistant Staph aureus or VRSA. There are still some antibiotics that are shown to be effective against MRSA, and those would be things like rifampin and Bactrim. But it's also led to the research into vaccines, which would be used primarily in folks who are high risk or have high susceptibility to getting MRSA infections, which, fun fact, would primarily be folks who are regularly in the hospital or in the hospitals for a long amount of time, as that's one of the most common places to pick up a Staph aureus infection, and that is Staphylococcus aureus.